This presentation is part of the Adverse Drug Reactions module, and it will address the question, what is pharmacovigilance? At the end of this presentation, you should be able to explain why the adverse drug reaction profile of a medicine remains unclear when it is given a marketing authorization, define the term pharmacovigilance, explain the ways in which adverse drug reactions to individual medicines can be identified, describe the responsibility of prescribers to contribute to pharmacovigilance processes, describe how to report a suspected adverse drug reaction via the yellow card system, and identify which adverse drug reactions should be reported. I would like to begin by considering why adverse drug reactions, or ADRs, are an important consideration in modern healthcare. All medicines have the potential to cause harmful or unwanted effects, as well as the beneficial effects for which they have been prescribed. Prescribers have to make judgments about the likelihood of each outcome before writing a prescription. For this reason, they need to be as well informed as possible about the potential adverse drug reactions associated with individual drugs. Although many adverse drug reactions are recognized during drug development, others may not emerge until later in the life of a marketed drug. This highlights the importance of having mechanisms in place that allow regulators to review the safety of medicines so that the balance between benefit and harm can continually be reassessed. Adverse drug reactions are a common clinical problem and will be encountered by healthcare professionals wherever they work in the health service. They account for around 7% of unscheduled or emergency admissions to hospital. They affect around 15% of hospital inpatients. They are the cause for around 3% of consultations in primary care. Careful retrospective analysis indicates that many of these episodes could have been avoided with more judicious prescribing and follow-up. Common culprit drugs are diuretics, anticoagulants, non-steroidal anti-inflammatory drugs, opioid analgesics, and insulin. More widely, adverse drug reactions may mimic the symptoms and signs of other diseases and cause diagnostic confusion. They may also affect the quality of life and tend to erode confidence in healthcare generally. In some cases, medicines may have a greater impact on the patient than the symptoms of the disease itself. For instance, drugs used to treat hypertension often produce adverse effects, while hypertension itself is an asymptomatic condition. And adverse drug reactions may reduce adherence rates and potentially delay the successful treatment of the primary clinical condition. Why is it that, in spite of the massive expenditure involved in bringing a new drug to the market, that some adverse drug reactions still remain undetected at launch? New medicines are approved for use on the basis of preclinical testing involving animal studies, phase 1 studies in healthy volunteers, phase 2 studies to explore the pharmacokinetic and pharmacodynamic profile in small numbers of patients, and then phase 3 confirmatory studies in a larger number of patients. At this point, the manufacturer will have collected sufficient data to make an application for marketing authorization to the regulatory bodies. However, the clinical trials they have undertaken may only have exposed 1,000 patients to the new medicine. Although any ADRs affecting the participants will have been carefully recorded, such trials are unlikely to detect all of the important adverse drug reactions because patients selected for the clinical trials used in the drug development process are carefully screened and may not reflect the real-world population that will subsequently be exposed. Notably, they rarely include children, women of childbearing potential, and the very elderly. They also tend to exclude patients with multiple comorbidities taking other medicines. Few trials used for licensing will have sufficient numbers of participants to detect rare ADRs. In other words, they are underpowered. Clinical trials are less likely to reveal ADRs that occur sometime after exposure, for instance myocardial damage occurring sometime after a relatively short-term course of anthracyclines for childhood cancer, or result from long-term treatment, for instance bone fractures after long-term use of some anticonvulsants. Some adverse drug reactions, even if they are common, may not be considered as having been caused by a drug if they are common events in the relevant study population. For instance, myocardial infarction is very common in patients with diabetes. For these reasons, less common adverse drug reactions can only be detected if there are systems in place to continuously monitor potential ADRs occurring amongst the many thousands of patients who are exposed post-authorization. So what is the definition of pharmacovigilance? The World Health Organization defines pharmacovigilance as the science and activities relating to the detection, assessment, understanding and prevention of adverse effects or any other drug-related problems. The aim of pharmacovigilance is to ensure that, as far as possible, drugs are selected and prescribed, taking full account of up-to-date knowledge of the harms they might cause. Pharmacovigilance is about protecting patients by enabling a more accurate assessment of risk versus benefit to be made, providing better information for prescribers and patients, and if necessary, withdrawing or restricting the use of medicines. Within that general framework of activities, pharmacovigilance can examine various aspects of adverse reactions. Most fundamentally, it uses various methods to identify evidence of previously unrecognized ADRs, a process often termed signal generation. It may also identify predisposing factors for known or previously unknown reactions, such as renal failure predisposing to lactic acidosis with metformin, or the increased likelihood of myopathies in Asian patients taking rosuvastatin. There may also be an opportunity to highlight differences in safety between two drugs in the same class, such as the likelihood of developing the metabolic syndrome with different atypical antipsychotics. After a signal has been generated, it undergoes careful review by the regulatory bodies. The outcome of the review may lead to various different kinds of action. First, there may be no action taken if the signal is considered weak or insignificant in terms of patient safety. There may be a new or strengthened warning put in place, which may be communicated in the product information and Dear Healthcare Professional letters. In the UK, warnings may be disseminated in Drug Safety Update and the British National Formulary. It may be possible to put in place new risk minimisation measures that minimise the chances of an ADR or the opportunity for vulnerable patients to be exposed to that risk. The legal status of a medicine might be changed. For example, a pharmacy medicine may be changed to a prescription-only medicine, meaning that it can no longer be made available without a prescription. Finally, the marketing authorization of a medicine may be suspended pending further evidence or revoked permanently. Who are the stakeholders involved in the pharmacovigilance process? All healthcare professionals have a duty to improve the safety of medical care for patients and can do so by contributing to pharmacovigilance activities. They can play this role in two ways. First, they can make reports via spontaneous ADR reporting systems, such as the Yellow Card scheme in the UK. Second, they might contribute to active surveillance systems, such as prescription event monitoring. For doctors in the UK, the importance of these activities are highlighted by the General Medical Council in its guidance Good Medical Practice. Healthcare professionals are often best placed to identify an untoward event, such as a suspected ADR to one or more medicines. Reporting such events can allow regulators to fully assess their significance and to act on the findings. Pharmaceutical companies are required to have a system for pharmacovigilance and risk management to make sure that they can take responsibility for their products and act appropriately when any risk is identified. They are obliged to pass on reports of adverse reactions to appropriate medicines regulators. Regulatory bodies such as the Medicines and Healthcare Products Regulatory Agency, or MHRA in the UK, the European Medicines Agency and the Food and Drug Administration in the US have major responsibility for pharmacovigilance. Their activities include operating spontaneous reporting schemes such as the Yellow Card scheme, monitoring clinical literature for information on potential harms from medicines, assessing reports of suspected ADRs, using its regulatory powers to ensure that the marketing authorizations reflect current knowledge and potential harm, and communicating with healthcare professionals about new information on risks and how they should be managed. Lastly, a stakeholder group of increasing importance are the general public, including patients and carers. Although formerly little involved in pharmacovigilance, reporting schemes are now actively inviting patients and carers to report adverse events suspected to be related to medicines. Current studies suggest that these reports have similar value to those provided by healthcare professionals. Let's now turn to the kind of methods of pharmacovigilance that are in common use in order to detect signals of adverse drug reactions. There are several ways in which adverse drug reactions to new or established drugs might be detected. Many of the more common adverse drug reactions will come to light during the drug development process. The testing of drugs using animals in the early preclinical stage of development might highlight potential problems that might be relevant to patients, for instance abnormal liver function tests. This will often preclude further development because of the risks involved. Phase 1 to 3 clinical studies in healthy volunteers and patients will highlight common ADRs, but the small number and careful selection of participants means that rarer ADRs may not emerge until the drug is used more widely in the real world. Once a medicine is licensed and marketed, many of the common ADRs have been identified, and it starts to be used in the much less closely monitored and variable supervision of ordinary clinical practice. Different pharmacovigilance methods must then be used to detect rarer but potentially serious adverse reactions. Voluntary reporting systems such as the UK Yellow Card scheme run by the MHRA are available to collect spontaneous reports of suspected reactions, usually from healthcare professionals, and enter them into a single database that can be examined for signals of previously unrecognised ADRs. Prescription event monitoring schemes identify patients exposed to a drug based on pharmacy dispensing records and follow them up for any subsequent medical events. Observational cohort studies observe populations or registries of patients and aim to detect events which might be the result of a drug. Patients experiencing the event are compared with unaffected controls, and the exposure of each group to potential culprit drugs is compared. Population statistics use record linkage to link together large electronic databases of prescription and healthcare events to search for signals that relate drug exposure to illness. Large Phase 4 clinical studies of licensed drugs, perhaps organised to compare one drug with another, are an important opportunity to identify new signals of less common reactions if they contain sufficient numbers of patients. The medical literature is another important opportunity to identify new drug safety concerns from case reports or small case series. A new opportunity that is emerging is the potential use of social media reports to generate signals relating to drug safety. I would now like to review some of the specific methods of pharmacovigilance in more detail, beginning with voluntary reporting schemes. In 1958, the drug Distaval was marketed by Distillers Limited as a mild hypnotic on the basis of its comparative safety over the alternative use of barbiturates. The advert depicts a child reaching into the medicines cabinet and suggests that this child's life may depend on the safety of Distaval, with the implication that the toll of barbiturate-related poisoning and death might be avoided. We all now recognise the medicine Distaval better by the name of its major generic component, thalidomide. Shortly after its marketing, a dramatic increase was seen in the frequency of a previously rare birth defect, phocomelia, the absence of limbs or parts of limbs. Unfortunately, it took 3 years to collect enough case reports to establish the cause as in utero exposure to thalidomide. In the United Kingdom, the response to the thalidomide disaster was to set up the Committee on Safety of Drugs under the chairmanship of Sir Derek Dunlop to develop mechanisms to ensure that a similar accident couldn't happen again. The committee had two main functions. First, to ensure that drug safety was adequately assessed before marketing. prior to the thalidomide disaster, any drug manufacturer could market any product in the UK without the need to satisfy any independent body with regard to its efficacy and safety. Second, the committee aimed to establish a voluntary reporting mechanism which came to be called the Yellow Card Scheme, which invited all doctors to report suspected adverse drug reactions using special yellow cards. The subsequent 1968 Medicines Act further enshrined pharmacovigilance as a key activity of the Committee on Safety of Medicines within the Medicines Control Agency. These functions are now served by the Commission on Human Medicines within the Medicines and Healthcare Products Regulatory Agency, or MHRA. After a relatively slow start, the number of reports began to increase rapidly. Factors involved in this increase have been greater awareness of the scheme amongst healthcare professionals, increasing numbers of doctors, more medicines available to prescribe, more prescriptions being written, now amounting to over 1 billion annually in the UK, a more elderly and sick patient cohort that was more vulnerable to the adverse effects of medicines, the availability of electronic reporting methods increasingly linked to electronic health records, making it easier to file a report. When the Yellow Card Scheme was first introduced, only doctors and dentists could submit reports. This has been gradually extended, and now all healthcare professionals, including coroners, hospital pharmacists from 1997, community pharmacists from 1999, nurses, midwives and health visitors from 2002, are able to report adverse drug reactions via the scheme. Other non-medical prescribers, including radiographers and optometrists, are now also encouraged to use the Yellow Card Scheme. Pilot studies in each case were able to demonstrate that the quality of reports coming from these professional groups was as good as the other professional groups already reporting. Since 2008, patients and their carers have been encouraged to provide voluntary reports of ADRs. Similar studies have demonstrated that their reports are as valuable to the signal generation process as those from the healthcare professional groups. In contrast to these voluntary reporting groups, pharmaceutical companies have a legal obligation to report any adverse drug reactions that come to their attention. They currently contribute nearly half of all reports to the regulatory agencies. Doctors, especially GPs, have formerly been the largest professional reporting groups, although other groups are now playing a more dominant role, with patient reports being the largest growth area. For many years, the Yellow Card has been available in paper version in the back of the British National Formulary or the ABPI Medicines Compendium. This mechanism of reporting has been superseded by online electronic reporting using either the MHRA website or more recently developed Yellow Card app for smartphones and tablets. There are 4 important pieces of information that must be included on each report. First, it is important to clarify the name of the suspected medicine, including brand and batch number, if known, the route of administration, the daily dose, the date the medicine started and stopped if applicable, and the reason why the medication was given. Multiple drugs can be listed if more than one drug is suspected of causing the reaction. Second, the suspected reaction must be carefully described with a diagnosis if known, when the reaction occurred, whether it was serious or not, any treatment given, and the eventual outcome. Third, required patient details will include age and sex, and some identification that will make the patient known to the reporter in the event of any future correspondence. The patient's name or date of birth are not required to protect confidentiality. Finally, the reporter must provide their own details so that their report can be acknowledged and follow-up information be obtained if necessary. In past times, when paper reporting was the norm, providing this information could be a time-consuming task. However, the advent of electronic reporting will mean that in future, the process will be facilitated by e-health records that can be used to immediately populate the e-yellow card, allowing reports to be generated and transmitted more quickly. Some reporters are unclear about which reactions they should report. Reporters are encouraged to consider reporting the following suspected reactions. Regardless of how long a drug has been on the market, any serious reaction thought to be associated with a medicine or vaccine should be reported. Serious reactions are those that are fatal, life-threatening, incapacitating, or that require hospitalization. The MHRA's surveillance of adverse effects of new drugs is more intensive. For such medicines, an inverted black triangle is shown against the name of the medicine in the summary of product characteristics and in the British National Formulary. Any adverse event suspected to be associated with black triangle drugs should be reported. Drugs are under intensive surveillance typically for a couple of years, after which the black triangle denotation is removed. If there is a doubt about the risk profile of a medicine, then the black triangle status is retained for longer or sometimes reinstated. All suspected adverse reactions in children should be reported, whether from established medicines or newly launched ones, whether from off-label use or from licensed use. For established drugs without black triangle status, there is no need to report well-known non-serious adverse effects, such as low-grade fever with vaccines or constipation with opioid analgesics. Finally, it is not necessary to be certain that the drug has caused the adverse drug reaction before reporting. A suspicion that an adverse event may have been caused by a drug is sufficient. If in doubt, report. After a yellow card is received by the MHRA, the data is entered into the database and the reporter is acknowledged. Further information might be requested. There is then a series of steps in the analysis of the data before it is batched and regularly placed in front of the Commission on Human Medicines. Particularly important amongst those steps is an assessment of causality. Causality is assessed based upon the nature of the reaction, its timing, the relation of the reaction to dose, consideration of other possible causes for the symptoms that have been attributed to drug exposure, evidence of improvement when the drug is stopped, previous reports of similar reactions to this drug, and, occasionally, data that suggest return of the symptoms on re-challenge with the same drug. Based on these considerations, the Commission on Human Medicines evaluates the strength of the evidence, the public health impact of newly identified risks, and any change to the risk-benefit balance of the medicine. On the basis of this assessment, it might recommend changes to prescribing information in the SPC, for instance restriction in use, reduction in dose, special warnings and precautions. It might recommend providing information and warnings to prescribers, or, in some cases, withdrawal of a drug if patient safety is threatened. The yellow card scheme has served UK pharmacovigilance well over 5 decades and has important strengths in being able to provide continuous surveillance during the whole lifetime of a product on the market and acting as an early warning system that generates new safety signals. In spite of the development of new methodologies, spontaneous reporting remains a vital part of the pharmacovigilance process. The yellow card scheme has led to many important regulatory changes, some of which are listed here. There are nevertheless several important weaknesses of the yellow card scheme and other spontaneous reporting systems. Overall reporting rates are very low as a proportion of all the adverse events occurring. As few as 4% of all ADRs are reported, and possibly only 10% of those that might be classed as serious. Reporting depends on awareness and motivation. Unfortunately, many healthcare professionals are unaware, often very busy, and some may not be motivated to take part. Reporting has to begin with recognising that an ADR might have occurred, and sometimes this may be missed. Reporting rate is high early after launch of a new drug, but vigilance and reporting then falls off rapidly. Unfortunately, reporting rate is heavily influenced by publicity, and reports tend to arise after a drug reaction is established, which, although valuable, are not as important as helping to detect signals of previously unknown reactions. Although spontaneous reporting can help to generate signals, it can't quantify the extent of any risk because not all reactions are reported and the total number and duration of exposures to the drug in question is also unknown. These inherent and important weaknesses have stimulated interest in other approaches to pharmacovigilance, and I would now like to consider some of them. Unlike clinical trials, which involve a specific intervention, observational studies observe what happens in the real world of clinical practice and try to infer relationships between specific exposures, including drugs, and subsequent clinical events. They are a major part of the discipline of pharmacoepidemiology, which concerns the use and impact of medicines in the population. Observational studies can very simply be divided into those that are prospective and those that are retrospective. Prominent examples of retrospective research are case control studies, which identify patient cases in the population that have had the clinical event of interest, and then compare them with matched controls who are event-free. The researchers then look retrospectively to see if there are any differences in exposure to potential causative drugs in the past. An example of prospective research is the cohort study, where a specific cohort of the population, free from the outcome of relevance, is identified and then followed up into the future. During this follow-up, events of interest will emerge, but the majority of the cohort will remain event-free. The researchers can then compare the exposure of each group to drugs of interest during the follow-up period and calculate how likely it is that the events were related to drug exposure. A major weakness of observational studies is confounding. This describes the potential for an association between a drug and an event to occur for reasons other than causation. There may be confounding factors that mean that patients who go on to develop events are more likely to be prescribed the drug of interest. For instance, if two drugs, A and B, are indicated for the same condition, but drug B is relatively contraindicated in patients with heart failure, then more patients with heart failure are likely to be prescribed drug A in routine clinical practice than drug B. Since heart failure carries a poor prognosis, there may be more patients who are exposed to drug A who suffer a cardiac death than those exposed to drug B. Researchers try to control for these factors if they are known, but there remains the possibility of remaining unknown confounding factors. Registry studies are a particular kind of prospective cohort study based on specific groups, characterized either by the clinical condition they have or the drug to which they are exposed. Unlike routine cohort studies, registries involve participants who have been very carefully characterized at entry to the study and who are then followed up with systematically collected data from real-world clinical practice. Another important pharmacoepidemiological approach is to look more widely at routinely collected health records that contain records of clinically important events and prescribing patterns. The term record linkage describes a systematic process by which the healthcare records of individuals in a large population can be brought together. This is often a major challenge because medical data arising from different healthcare contacts are usually stored separately. There are also other important challenges. In order to make the record linkages, there have to be patient identifiers that enable separately held data for an individual to be brought together. The sharing of these identifiers raises significant privacy issues. Another hurdle is having sufficient computing power to undertake matching and look for signals involving billions of data points, although this has been facilitated in recent years by advances in computerization. Record linkage has the potential to provide real-time, continuous surveillance for signals of adverse drug reactions, taking almost all health data into account. No longer will the regulators be dependent on what they are told by a small number of enthusiastic reporters. A number of large organisations are already established and doing this work. In the UK, the relevant organisation is the Clinical Practice Research Database, or CPRD. It should be noted that, like all observational data, record linkage studies have to consider the potential for confounding when interpreting their results. There are various other methods of pharmacovigilance. After a drug has been licensed and marketed, large Phase 4 clinical trials, sometimes involving thousands of patients, may be undertaken to compare the new drug with existing alternatives to discover whether there might be a clinical benefit. Although these studies have the potential to highlight important ADRs associated with a drug and quantify their frequency, often expressed as numbers needed to harm or NNH, they have some important drawbacks. The trials usually include homogeneous sample populations with little of the variation that would be found in the real world. Few of the patients have significant comorbidities. Few are taking many potential interacting drugs. Most trials exclude specific groups such as pregnant women, children, and elderly people. The sample size may be too small to detect relatively rare events. The trials are often of limited duration, precluding the discovery of long-term consequences such as cancer. Lastly, clinical trials are usually conducted in very controlled conditions and are unable to predict the real world. Reviewing the medical literature for case reports or case series that describe adverse drug reactions and other drug safety issues can sometimes contribute to signal generation. The reports can then be compared with other data collections such as voluntary reports and help to build a clearer picture about previously unrecognized adverse reactions. Social media is rapidly changing the way we all communicate and may be set to change the face of signal generation in pharmacovigilance. In the UK, there are 30 million Facebook users and 15 million Twitter users, mostly amongst the younger age groups. Use of medicines and drugs form an important minority of these communications and is clearly presenting an opportunity to detect new issues at an early stage in their development. There are obvious challenges in being able to decipher these often very brief messages potentially containing spelling errors. However, some studies are offering the hope that the frequency of reports of adverse events related to medicinal products detected in this way seem to match reports collected from more conventional routes. The MHRA is currently part of a multi-stakeholder research consortium called Web Radar that is investigating the potential power of this most modern of approaches to pharmacovigilance. That brings me to a brief review of the key points of this presentation. Information on the potential hazards associated with a new drug is incomplete when it is launched because of the relatively small numbers of patients that have been exposed. Pharmacovigilance is the process of monitoring the safety of drugs after they have been approved for use by the regulatory bodies. Healthcare professionals, pharmaceutical companies and regulators all play a role in pharmacovigilance. The main approaches to pharmacovigilance include spontaneous reporting of adverse drug reactions by healthcare professionals and patients, pharmacoepidemiological studies, and record linkage studies. You can find further reading and resources to support your learning at the locations above. I hope you found this presentation helpful and look forward to you joining me again for more clinical pharmacology shortly.